Hi, my name is Simon Paquette, and today I'll be talking about genetically engineered microbial Lactobacillus reuteri as mediator for radiation therapy-induced bowel injury through the upregulation of reuterin. I have no affiliations or relationships to disclose, and this research received funding from the New Frontiers in Research Fund and the Natural Sciences and Engineering Research Council of Canada. Starting off with a little bit of background, the gastrointestinal tract is particularly significant in radiation therapy due to its sensitivity to radiation-induced damage. The ability of radiation therapy to treat tumors is disadvantaged by negative side effects on healthy tissues. These side effects can cause acute symptoms that can lead to discontinuation of the radiation therapy regime and cause significant morbidity. To date, mitigation strategies have focused on methods that increase gastrointestinal tissue resistance to radiation. However, this strategy has been studied for decades, yet there is still no effective treatment options. An overlooked component of the pathophysiology of radiation-induced bowel injury is the role of the gut microbiome. Both the gut and the microbiota provide bidirectional beneficial signals, resulting in a symbiotic environment for optimal gut health. The microbiome plays a vital role in protecting the gut from injury by releasing metabolites that aid in the recovery of injured gastrointestinal tissue. L. reuteri, a gram-positive and rod-shaped bacterium, produces 3-HPA or reuterin when in anaerobic conditions. The reuterin system is active against gram-positive and gram-negative bacteria, as well as yeast, molds, and protozoa. Reuterin's precursor is glycerol and is converted intracellularly thanks to an enzyme made up of 3 genes of the PDU-CDE. When reuterin is dehydrated to acrolein, it forms adducts with cellular targets such as proteins and DNA and disrupts multiple biochemical pathways. Thus, it may help prevent radiation-induced bowel injury and accelerate recovery. We can look at Figure 1 to see all of the processes. As for the methods, fully sequenced L. reuteri was grown anaerobically using lactobacilli, MRS, agar, and broth. Figure 2 is an overview of the genetic engineering. The plasmid PBS LDH GFP was excised from E. coli using MediPrep. A restriction digest was performed to prepare the backbone and insert for ligation. GFP was then excised as a result from our plasmid. Samples were separated through gel electrophoresis, and the appropriate sections of the agarose gel were excised and gel purified to obtain the backbone and the insert for the ligation. The PDU-CDE and the vector were ligated using T4 DNA ligase. The product was inserted or transformed chemically into competent E. coli on LB-agar plates with ampicillin, and the clones with the insert were verified using the colony PCR. Our next step would be to take the mutant plasmid and introduce them into L. reuteri cells by electroporation. We will verify the gene expression using quantitative PCR, and we will also verify that reuterin is upregulated by performing a colorimetric assay or a high-performance liquid chromatography. As for the results, Figure 4 demonstrates the L. reuteri anaerobic growth curve, and we also have the results in Figure 5 of the gel electrophoresis as the colony PCR. Thank you so much.